In this video, I'm going to discuss some key primary immunodeficiencies as well as some of the disease susceptibilities associated with them. And this should prepare you for the TBL that we will have next week. Now, I've given you a table in your TBL of a number of primary immunodeficiencies that you can study on your own, but those other than the ones listed here, those primary immunodeficiencies will be outside the scope of this TBL. In this video, I'm only going to focus on the key mechanisms of these primary immunodeficiencies. And so for the purposes of the IRAT and for the TBL, it will be important for you to be able to describe the mechanism of action of these immunodeficiencies, as well as the disease susceptibilities associated with them. So first, we're going to start with severe combined immunodeficiency, or SCID. This is a simultaneous loss of B and T cell function. Now, this is just meant to be a picture of a B cell lacking antibody responses or a T cell lacking the ability to make cytokines or exert other effector functions. And this is often a disease that results from defects in early lymphocyte development. And people with SCID have severe disease susceptibilities that start at birth. Often this can include opportunistic infections, and many of the disease susceptibilities for SCID actually overlap with T cell deficiencies because T cell deficiencies also result in susceptibility to opportunistic infection. Infants with SCID exhibit a failure to thrive, and this is a fatal disease if untreated, and so hematic stem cell transplantation is the preferred method as early as possible to treat this disease, although there are some gene therapy methods that are being studied as potential alternative approaches. The most common form of SCID is the X-linked version, and this is due to a defect in a cytokine receptor called the interleukin-2 receptor gamma. Now, on the left here, I've showed an activated T cell that expresses the interleukin-2 receptor. Interleukin-2 is a cytokine that induces T cell clonal expansion, and the receptor is composed of 3 subunits. This gamma subunit is actually sometimes referred to as the common gamma chain because it is a subunit of multiple different cytokine receptors. And one of those cytokine receptors is the interleukin-7 receptor. And for the purposes of X-linked SCID, it is the inability to respond to interleukin-7 that is important. Interleukin-7 is a cytokine that drives the formation of common lymphoid progenitors as well as their survival and proliferation. This interleukin-2 receptor gamma forms part of this interleukin-7 receptor overall, and therefore the defect in X-linked SCID can most accurately be described as a defect in the interleukin-7 receptor. In the absence of this common gamma chain, this IL-2 receptor gamma, developing lymphocytes in the bone marrow can no longer respond to interleukin-7, and this leads to an absence of T and NK cells. In these patients, B cells may be present or even increased, but they can't make any immunoglobulin. And so these are individuals that have combined defects in both T cell and B cell function. There are autosomal recessive versions of SCID. I'm only going to talk about one of them, and that is SCID that results from a loss of one of these 2 enzymes, either purine nucleoside phosphorylase, PNP, or adenosine deaminase, or ADA. And these are enzymes that are important in clearing toxic metabolites from cells. In the absence of PNP and ADA, these toxic metabolites cannot be cleared. And because developing lymphocytes are among the most proliferative cells in the body, this particularly affects those cell types, including B cells, T cells, and natural killer cells. This leads to a progressive loss of adaptive immune function. Now, we screen now for SCID. In fact, newborn screening for SCID as of 2019 is done in all 50 states. The most common way to screen for SCID is through an assay called a TREC assay. TREC stands for T cell receptor excision circle. This is a circular DNA structure that is generated during lymphocyte rearrangement within the bone marrow, and also created when T cells mature in the thymus. This is an easy structure to detect and actually can be detected using a dried blood spot. There are some life-saving interventions when a baby is found to have SCID. One is to stop breastfeeding right away because this will decrease the transmission of CMV, which can cause severe disease. The other is to perform an early hematopoietic stem cell transplant. And lastly, there are some enzyme replacement therapies, particularly for the ADA version of SCID. Moving on to antibody deficiencies, one of the key things to know about antibody deficiencies is that unlike SCID, the onset of disease susceptibility won't usually start until after 4 to 6 months after birth. And the reason for this is that's about the time that maternal antibodies wane. This can result in recurrent bacterial infections, again, unexpected frequency and severity or unexpected complications. Often these are infections of the respiratory tract, including sinusitis and otitis, and often can be the result of unexpected vaccine preventable infections, such as, for example, Haemophilus influenza. The most common antibody deficiencies are combined variable immunodeficiency and IgA deficiency. CVID actually doesn't onset typically in very young individuals, but onsets later in the second or third decade. And the genetic basis for this is really unclear. For most cases, it's actually unknown, but can result from any defect in B cell survival or defects in B cell activation through, for example, co-stimulation. And CVID is characterized by variable defects in antibody production. It can be a defect in one isotype, it can be a defect in all isotypes, and often results in recurrent and chronic infections. IgA deficiency is the most common primary immunodeficiency, and in fact, 80 to 90% of people with this deficiency are asymptomatic. But if you are symptomatic, this can result in recurrent infections, chronic diarrhea, for example, giardiasis, and this is a disease that can progress to CVID and also is linked to an increased risk of autoimmune disease. The next antibody deficiency that I want to just discuss briefly is X-linked A-gamma globulinemia. This is a disease that results from defective cell signaling during lymphocyte development. Now, under normal circumstances, when our antibody receptors rearrange, and the heavy chain rearranges successfully, it gets expressed at the cell surface along with this companion molecule, the pre-B cell receptor that stabilizes its surface expression. When this happens, this sends a survival signal into the cell that tells the cell that a successful rearrangement has occurred. And one of the key molecules that mediates this survival signal is Bruton's tyrosine kinase, which also happens to be expressed on the X chromosome. In the absence of successful heavy chain rearrangement, developing lymphocytes undergo apoptosis by default. In the absence of Bruton's tyrosine kinase, even when heavy chain rearrangement is successful, the cell has no way to signal to itself that it has undergone a successful heavy chain rearrangement because the signaling pathway is now impaired due to the absence of Bruton's tyrosine kinase. This leads to a disease called X-linked A-gamma globulinemia and leads to apoptosis of all developing B cells. So no mature B cells, no immunoglobulin, you get these recurrent infections and can be susceptible to chronic enteroviral meningoencephalitis. And the last antibody deficiency I wanted to mention to you is hyper IgM syndrome. This results from the inability of a T cell to provide adequate help to a B cell to induce it to form a germinal center. And this is because the deficiency in hyper IgM syndrome is an inability to express this co-stimulatory molecule called CD40 ligand, which again is expressed on the X chromosome. CD40 ligand binds to CD40 on B cells and induces the formation of germinal centers and isotype class switching. The loss of this molecule means no germinal center and no isotype class switching, meaning that whereas B cells can get activated, they're only going to be making lots and lots of IgM. They're unable to make other isotypes like IgG. Individuals with hyper IgM syndrome often experience neutropenia for unknown reasons. They have a similar susceptibility to severe bacterial infection as other IgE deficiencies, but they also seem to be susceptible to some opportunistic infections such as pneumocystis jirovecii pneumonia. And that is likely due to because the defect in CD40 ligand not only impacts B cell activation, but also impairs certain T cell protective functions. Moving now to T cell deficiencies, I'm only going to mention one, and that is DiGeorge syndrome. DiGeorge syndrome results from a chromosomal deletion in chromosome 22 and can result in either a partial or complete failure of thymic development. Partial DiGeorge may present some T cell defects, but complete DiGeorge, which happens in only about 1% of cases, can result in the absence of T cells altogether. Now, the important thing to know about DiGeorge, however, is that there are a number of other defects associated with DiGeorge because it is a chromosomal deletion. This includes hypoparathyroidism and hypocalcemia, along with congenital heart defects and some cranial and facial deformities, such as cleft palate and rounded jaw, as shown in these examples on the left. Individuals with DiGeorge syndrome can be susceptible to severe infections as well as opportunistic infections. And really the only solution for an individual with complete DiGeorge is to do a thymic transplant. Now, there are some other congenital deficiencies that do impact the adaptive response. For the purposes of this TBL, we're kind of skipping over these deficiencies. I'm not going to test you on them. I'm not going to hold you responsible for them for now. But I do want to make you aware of them because they may come up on boards. And one of those is called Wiskott-Aldrich syndrome. This is a defect in cytoskeletal structure that impacts T cell activation and migration. Another is ataxia telangiectasia. This is a defect in the ATM kinase, which repairs double-stranded DNA breaks. This leads to defects in lymphocyte development because, as you remember, all lymphocytes undergo DNA rearrangement to generate their antigen receptor. There are some defects in certain cytokines that lead to specific susceptibilities to disease. For example, individuals with defects in the ability to produce the cytokine interleukin 17 can be susceptible to fungal infection, including chronic candidiasis. There are other defects in things like transcription factors that can impair the ability of the immune system to make effector T cell subsets. So, for example, in hyper IgE syndrome or Job syndrome, individuals lack the ability to make STAT3. And in the absence of this transcription factor, this results in overactive Th2 responses and a loss of Th1 and Th17 responses. Now, what this means is that you can drive immune responses that can be protective against some types of infections, but not others. And lastly, there are known defects in the expression of MHC class 1 and MHC class 2. Okay. Now, I'm going to move now to immunodeficiencies of the innate immune system. I'm going to largely focus on immunodeficiencies of the innate immune response. I'm going to focus on chronic granulomatous disease and leukocyte adhesion deficiency. And then I'm also going to talk briefly about some immune deficiencies related to complement. So first of all, complement deficiencies of either the classical pathway or the alternative pathway or the membrane attack complex or even inhibitors of complement all result in the same or susceptibility to infection, and that is susceptibility to infection with Neisseria meningitidis. Now, that you can also have infection with other encapsulated bacteria. The two other main culprits are Streptococcus pneumoniae and Haemophilus influenza. But when you think of complement deficiency, the main infection that you're thinking of is Neisseria meningitidis. Now, you may wonder why inhibitors of complement lead to susceptibility with Neisseria meningitidis. Shouldn't complement work better when these inhibitors are absent? But what ends up happening is when these inhibitors are absent, complement becomes overactive, and because of this overactivity, becomes defective. It consumes all of the complement components that it needs to form a normal induced response to infection. Now, one of the interesting things about complement is that there is an antibody, eculizumab, that can be used that targets the terminal complement complex or the membrane attack complex. And this is used for atypical hemolytic uremic syndrome, as well as in certain transplant settings. And because it targets this terminal complement, it actually leads to susceptibility to Neisseria meningitis, just like the primary complement immunodeficiencies do. The intervention here is to vaccinate against meningococcal bacteria at least 2 weeks before starting treatment with this drug. And antibody prophylaxis as well is often used. Now, this is not a primary immunodeficiency. This could most accurately be described as an acquired immunodeficiency. I just wanted to present it here so that I could indicate that any interruption of this pathway can lead to similar disease susceptibilities. There are 2 diseases that I want to briefly mention that are associated with defects in complement pathways. One of them is called paroxysmal nocturnal hemoglobinuria, or PNH. This is complement-induced intravascular hemolytic anemia that often results in dark urine in the morning and thrombosis. It results from a defect in PIG-A. This is an enzyme that induces the formation of GPI anchors for some membrane-associated proteins, including 2 complement inhibitors, CD59 and DAF. And in the absence of these inhibitors, individuals are susceptible to periodic episodes of complement overactivity, resulting in red blood cell lysis and heightened susceptibility to Neisseria because the complement system doesn't function normally. This is actually a rare example of an acquired mutation, not an inherited mutation. And the other one that I wanted to mention briefly is hereditary angioedema, HAE. This also results from a defect in a complement component called C1 inhibitor, but in this case, it has nothing to do with complement, because whereas C1 inhibitor does regulate certain aspects of complement activation, it also regulates bradykinin release. And in the absence of this inhibitor, individuals are susceptible to swelling of the feet, limbs, face, larynx, trachea, etc. And this is a disease that can become more severe in late childhood and adolescence. Now I'm going to end with just talking briefly about 2 diseases. This should be review for you. One of them is a disease called chronic granulomatous disease. And chronic granulomatous disease results from defects in the ability of phagolysosomes to undergo oxidative burst. And in particular, it's a defect in this enzymatic complex called the NADPH oxidase. There is one component of this complex that is expressed on the X chromosome, and this is therefore the most common version of this disease. And the inability of NADPH oxidase to function results in a loss of redox reactivity and an inability to undergo oxidative burst within the phagolysosome. The resulting inability to control infection leads to the formation of granulomas and susceptibility to bacterial and fungal infections. And this is a picture of what a granuloma may often look like, which is a pathogen surrounded by a ring of infected macrophages, surrounded by a ring of T cells, and generally an inability to get rid of those pathogens. The second defect in the innate immune response that I want to mention briefly is called leukocyte adhesion deficiency, and this results from a loss of this molecule called LFA1. LFA1 is an integrin that mediates the adherence of leukocytes as they travel through endothelial cells to the endothelial barrier at sites of inflammation. This adherence, this firm adherence, is required for these leukocytes to stop and migrate across the endothelial barrier into tissues where they can then migrate to the site of infection. In the absence of LFA1, this never occurs, and so you'll end up with a raging infection into tissue, lots of production of inflammatory cytokines, but an inability of these leukocytes to be able to stop and adhere and migrate into those sites of infection. As a result, you'll end up with sites of infection with no leukocytes, but in the blood, you'll end up with huge numbers of neutrophils and an unresolved cytokine response. And this results in a susceptibility to severe bacterial infections. Okay, so that is just a quick review of some of the most important diseases that we would like you to know in advance of the TBL on Thursday.